On the other hand, the canned pineapple has to be heated to 100 degrees Celsius in order to destroy microorganisms that are able to grow without oxygen. This heating process also destroys the enzyme bromelain, as it can only withstand temperatures up to 70 degrees Celsius. Because the tinned can is heated past this point, the protein begins to denature and the enzyme is unable to function. Thus, when used to make Jello, bromelain is not catalyzing the hydrolysis of collagen. When the Jello was made using the canned pineapple, the Jello was able to form properly and solidify because the aqueous form of gelatin was able to reform bonds and trap the water molecules in between its chains effectively without the presence of the enzyme to break down the collagen. An experimental error in this experiment could be the temperature of the water. The temperature of the water used in the solution could have affected the results of the experiment. The instructions of the experiment say to use hot water from the kettle. However, there is no set temperature in the experiment for the heat of the water. For example, if boiling water was used to make the Jello, the heat would have increased the kinetic energy in the solution. This would have led to a faster reaction. The triple helix would have unraveled faster as the bonds would break quicker. This allows the water more time to enter in between the amino acid chains and for the bonds to reform around it. However, if warm water was used in comparison, the kinetic energy would be significantly less. The bonds would break slower, allowing less time for the water to become trapped between the amino acid chains. Using water at a cooler temperature would lead to a less solid Jello, versus at a boiling temperature, the reformation of the bonds would create an optimal semi-solid structure. Because the temperature from the cold water from the tap was also not regulated, there is no guarantee that the water from the tap was cold at all. In order for the gelatin to properly set, cool water is needed to regulate the temperature so that the hydrogen bonds of the gelatin chains begin to reform. However, if the tap produced warm water and that water was added to the solution, then the hydrogen bonds would not be able to reform properly, and then these would not be able to effectively trap the water in between the chains. In the long run, these effects are minuscule, as the main factor in this experiment is the presence of the bromelain protease enzyme. However, the temperature of the water may have contributed to deferred results. Overall, our hypothesis was proven correct, as the type of pineapple does affect the solidity of the Jello, and the fresh pineapple was able to create a more liquid Jello because the enzyme bromelain found in the pineapple catalyzed the protein collagen, breaking down the peptide bonds of the protein found in the Jello, and this denaturation led to the substance of the Jello to have a liquid state. In conclusion, it is safe to say that canned pineapple is best to use when making jelly to receive an optimal structure.